Hello friends, welcome to my channel Naved Biochemistry classes. Well, in this lecture I will give you introduction of splicing mediated by Spliceosome. Now, when you talk splicing of obviously splicing, when you're talking splicing of introns in eukaryotic mRNA precursor or primary mRNA transcript. So, splicing, where it takes place, in case of eukaryotic, mRNA precursor, it takes place in the nucleus. Either you say mRNA precursor, or you also called heterogeneous RNA, or you also call primary mRNA transcript. You also call it primary mRNA transcript. You can use either of the three. So, in case of eukaryotic mRNA precursor, it takes place in the nucleus. And since we are talking splicing mediated by spliceosomes, so what you have a spliceosomal intron. Spliceosomal intron. So obviously these are special introns. Why? And these are found. These are the largest group. These are the largest group in primary mRNA transcript in eukaryotic nucleus. Okay, so these two are the basics when we start splicing mediated by spliceosomes. And obviously when you talk mediated, you are saying that splicing is being catalyzed or intervened by spliceosome. Now we talk about a spliceosomal intron. So what is this all about, a spliceosomal intron? We got it that it is the largest intron. It is the largest intron. But why spliceosomal? Because these are spliceosomal intron. Well, these are intron which are removed by large protein complex which you call spliceosome. So here you got the name. So you have a special class of intron which will be removed only by this particular protein complex or large complex. It is made up of protein complex as well as RNA. We will talk it later. So now you see here what are the very interesting feature of spliceosome. Apart from that, it is a complex protein complex or a large complex molecule. Now, the interesting part is that ATP is required. ATP is required during its formation. When you say formation, obviously you are talking that it does not exist, it is formed. So when it is formed, it is formed during the process of transcription. During process of transcription. So it does not pre-exist as other cell organelles, rather it is formed. When it is formed, transcription, as you know, that is DNA to RNA, synthesis of DNA to RNA, and obviously what I have said and written also, that ATP is required during its formation. Now another interesting aspect of spliceosome is, now you say, I told you that spliceosomal intron are removed by this particular large protein complex spliceosomes. Now another interesting part, that apart from that it required ATP during its formation, it does not exist, and it is formed during the process of transcription. The other interesting is that splicing of this spliceosomal intron takes place in spliceosome itself. That is, all the process of this splicing will take place in this particular complex. What you call it? You call it spliceosome. Okay. Now, so this is all about spliceosome. Now we are telling that it is large. It is large. How large it is? It is, you can say that in size, it is as large as ribosome. So what is the upper and lower limit? Upper and lower limit is 3.5 megadalton to 4 megadalton in size. Okay. So when you talk Dalton, obviously you are talking 1.66 into 10 to the power minus 27 kg. Okay. So that is the atomic mass unit in terms of kg. So what is this? How size matters? So what is the function of its size? Now due to size, what happens? A spliceosome fits into nuclear pores. Okay, it fits into nuclear pores. So when you say size, now you are saying that it fits into nuclear pores. So what will happen? And now you are going. We have already talked that these splicing takes place in spliceosome itself. So what happens? Due to its size, it fits into nuclear pores. So what will happen? So what this phenomenon does, and what the size does. What it does, it prevents spliceosome prevents who? Prevents mRNA primary transcript escape from nucleus before its processing. So obviously, when you talk removal of a spliceosomal intron, it is one of the processing modification of the inactive mRNA apart from other I have told you. Okay. So this is the main main thing associated with the size. That is, it will fit the nuclear ports, the spliceosome, and prevents the mRNA, precursor mRNA, to escape from the nucleus before processing. Okay. Now, when we talk this that it is a large subunit containing proteins, so how it is made up? We told that it will be formed during transcription. So what is happening? Spliceosome is made up of around 45 to 50 proteins and 5 RNA. 5 RNA. Okay? And RNA is not long. It is up to made up of 500 to 100 nucleotides. So obviously, when you are talking 50 to 100. Obviously, it is a bit small. So these RNA, these 5 RNA, are called snRNA. So what is this? This is a small nuclear RNA, that is ribonucleic acid. So these RNA, the 5 RNA, you will call snRNA. Now another interesting is that they are designated. These are designated by some symbols. By, now you have 5. So you have U1, you have U2, you have U4, you have U5 and U6. Okay? You have U1, U2, U4, U5 and U6. Okay? So obviously you can see that U3 is missing. And as a student of biochemistry, you know when you talk you are talking either uracil or uridine. So this nomenclature is based on uridine content. Nomenclature is based on what? On uridine content. And when you talk uridine, obviously you are talking uracil and its ribose, combination of uracil and ribose. Now another thing is that these snRNA we have already told that the spliceosome is made up of 45 to 50 proteins and 5 RNA. We have now written about RNA. Now what happens, this snRNA that is associated with set of proteins, what we have already told, that is from out of 45 to 50. So obviously if you have snRNA plus protein, so what will be formed? You will be getting snRNPs. Okay? So you have snRNP, 1 minute, or you can also write snRNPs. So what it means, obviously we have already written snRNA means a small nuclear ribonucleic acid. So what it will be? snRNP will be a small nuclear ribonuclear protein. Now, these snRNPS is pronounced as SNRPs. It's pronounced as SNRPs. Okay? You don't say snRNP. You simply say SNRPs. Okay? So, now another interesting factor, if you are interested in history, well, this was discovered by Michael R. Lerner. Then you have another man and Sydney, two group of workers, and Sydney Altman. And also another fellow, another group, Thomas R. Sech and SECH and well, just for sorry, I have to make a correction here. It is not Sydney Altman. Actually, Sydney Altman belongs to the second group. This is Michael R. Lerner and John A. Status. Okay, and obviously these fellows were awarded Nobel Prize in 1989. Also chemistry Nobel Prize in 1989 they were awarded. Okay. So when we talk snRNA, you say that it is a small nuclear RNA. When you talk snRNP, SNRPs, obviously this is a small nuclear ribonuclear proteins. Now, one thing more. So it is obvious from these two, their relationship, you can say that snRNA is like ribosomes. snRNA in SNRPs is similar to ribosomes. It is similar to ribosomes. In which respect? Is similar to ribosomes. In which respects? In both as enzymatic, because snRNA in snRNP will be having both a structural and enzymatic role. Like enzymatic role like ribosomal RNA. Not ribosome, like ribosomal RNA. Okay? We already said that when you talk snRNA, they are associated with proteins to form SNRPs. Now, question is, you have only 5 snRNA, and what you say that you have 45 to 50 proteins. Okay. So, and now you are getting SNRs. So what happened in SNRs? So in SNRs, what let us in SNRs, one or two RNA. How much RNA? One or two. One or two RNA is there, and these are associated with a number of proteins. With maybe with one specific protein. Now let us talk more detail. One specific protein or a set of protein, or a set of protein. Okay. Now, so you have a set of protein. So what happened? One RNA. It is not that one RNA will have only one protein. One protein or set of protein can be shared by more than one RNA. So this is the thing that you have one or two RNA that can be associated with one specific or a set. Now again, one RNA can have two or more RNA can have same set of protein. That's the point I want to make it specific to you. That is, some protein can be common to all, and what happens while and some protein can only occur in one RNA. So all the things are open. So you already seen that finally you have snRNP, SNRs. So you can say that sorry, you have to always write this. When you talk SNRs, you have to, well, these are building block of Spliceosome. Well, these are building block of Spliceosomes, and obviously, since we said that Spliceosomes formation takes place during transcription, well, this Spliceosome formation takes place in steps. We will talk it formation of Spliceosome and its splicing mediated by Spliceosome when we talk its mechanism of the splicing mediated by Spliceosome. Right now we are only talking introduction related with your Spliceosome. So we have already said that the 5 RNA are U1, U2, U4, U5 and U6. But apart from that, there are other snRNA also found. Okay. So what are these? And these 5 RNA, that is 5 snRNA, that is U1, U2, U4, U5, U6, they are abundant in eukaryotic nucleus. Now, let us talk the other ones. You have now let us talk about this U3, U8 and U13. Obviously, we are talking its combination, that is 3 types in combination with protein, that is SNRs. So what you have, these are localized in nucleolus. It is not in nucleus. They are in nucleolus, that is part of nucleus, you know it. And these are not part of, that is, these are not take part in formation of spliceosome, but these are present. These SNRPs are present in, you can see, nucleolus, but these does not take part in formation of only five what I have designated already: U1, U2, U4, U5 and U6. Those are involved in formation of a spliceosome, but other are also present, and that is present and in nucleolus, and they do not take part in formation of. Apart from that, you have also U7. Okay, that is SNRNP U7, designated by U7, has also been found, and it is also not involved in splicing. It is also not involved in splicing. So how it is different from U3, U8 and U13? Well, it is responsible. It has some function which has been recognized. It is responsible for processing the 3' stem loop of what? Of histone pre-mRNA. So this is the function recognized for U7, that it is responsible for processing of the 3' stem loop of histone pre-mRNA. Obviously, when you talk loop and all these, you are talking some mechanisms. Since it is not part of a spliceosome, we will not go further detail. We will just giving some information which you may found interesting. And now another are also recognized, that is U11 and U12. So these are also interesting in the sense that they are required. Some splicing are U2 independent, and these are required where? In U2 SNRP independent splicing process. So what you have, you can see you have U1, U2, U4, U5, U6, that is the core snRNA, which is responsible for formation of a spliceosome, but it undergoes combination with protein to form SNRs. Now apart from that, I talk about the other one, that is U3, U8 and U13, which are present in nucleolus and does not take part in formation of a spliceosome. You have U7, but it is not involved in splicing, but it is responsible. You can see that in histone pre-mRNA 3 stem loop formation processing, it is involved. But if you have U1 and U12, some splicing is U2 independent. That is, you have U1, you have U4, you have U5, U6, but you don't have U2 in the process. So there you will require U1, U11 and U12 SNRPs as the building block of spliceosomes of different kind. Okay. So Another important aspect of a spliceosome, splicing mediated by spliceosome, is that a spliceosome, one of the components of a spliceosome, other component include SR protein. SR protein. Okay, so when you say SR, you are talking serine rich and arginine. Okay, so SR protein is SR protein rich. Obviously, you have, it is one of the important component in formation of spliceosome. Okay, and it is involved. This SR protein is involved in splicing during the process of splicing mediated by spliceosome. So this is an important one. That is, U11 is not involved. U3 is not involved, U8 is not involved, U13 is not involved, U11 and U12 are involved in UT independent, but you have SR protein which is involved in the formation of a spliceosome. It is important component of a spliceosome. Now the last is, when we talk SNRPS, SNRPs, okay, these are major autoantigen. These are major autoantigen. So what it means? It is major autoantigen in a spliceosome, and when you talk autoantigen, you are talking a normal or complex protein that is recognized by immune system of patients suffering from autoimmune disease. So that is the definition of autoantigen. So what autoantigen, you know that this is a major complex, it can be normal or it can be complex protein that is recognized by immune system of patients suffering from autoimmune disease. So this is all about introduction of spliceosome and splicing mediated by spliceosomes. I have given you some inputs. In the next lecture, I will give you the complete notes so that you can summarize it. Okay, thank you.